Assalamualaikum and hi. I am Adrianna Amani Binti Mohammad Ramdzan. This is my group partner.
Hi and Assalamualaikum. My name is Azra Hanis and I would like to present key findings from a scientific article titled Genome-Based Analysis of Klebsiella Species Isolates from Animals and Food Products in Germany 2013 to 2017. It is a study that highlights the genetic diversity, antibiotic resistance, and virulence factor of Klebsiella pneumonia and other Klebsiella species isolated from animal and food sources in Germany. This is an important topic as multi-drug resistant and virulent Klebsiella strain have become more significant threat to public health globally, yet the environmental sources and transmission route remain poorly understood. This study analyzed 94 Klebsiella species isolates from animals and food products between 2013 and 2017. The aim was to investigate the presence of antibiotic-resistant gene and virulent determinants such as YBT, CLB, IRO, IUC, and RMPA or A2. Researchers performed antibiotic susceptibility testing and used advanced whole genome sequencing (WGS) technique to understand the genetic profile of this isolate. The result revealed that around 77% of the isolate belong to the Klebsiella pneumonia sensulato complex with a high level of genetic diversity. 44 known sequence type, 18 new sequence type, and 38 capsule type were identified. Interestingly, ESBL genes which can confer resistance to many beta-lactam antibiotics were found in 16 isolates. Additionally, colistin resistance, a last resort antibiotic, was identified in one isolate. Virulence gene were present in 22 isolates and the aerobactin IUC gene was specifically linked to isolates from pigs and wild boars, which suggests that pig might act as a reservoir for certain virulent Klebsiella strain, though further research is needed to confirm this.
Klebsiella spp are gram-negative bacteria found in soil, water, animals, and also humans. The most clinically significant species is Klebsiella pneumonia, which causes pneumonia, bloodstream infections, and urinary tract infections, especially in immunocompromised individuals, meaning weak people. In recent years, multi-drug resistant and hypervirulent strains have emerged, posing major public health concerns. This study aimed to analyze the antibiotic resistance, virulence genes, and also the genetic diversity of Klebsiella isolates from animal and also food sources using whole genome sequencing.
Next, I will move to the methodology section, which underpins the reliability of these findings. So for the first one, the bacterial isolation and species identification process. A total of 90 main isolates were collected from animal and food sources by the Chemical and Veterinary Analysis Agency staggered between 2013 and 2017, with 4 reference isolates included for comparison. Initial species identification was performed using multi-toph mass spectrometry, which is a method that analyzes protein fingerprints of bacteria. However, multi-toph results can sometimes be limited when closely related species are involved, making a further genetic confirmation necessary. Second, whole genome sequencing and de novo assembly. Genomic DNA was extracted using a pure-link genomic DNA mini kit, ensuring a high-purity DNA for downstream applications. WGS was carried out using the Illumina MIC662 platform, which generates short-read data of high accuracy. For one particularly interesting isolate that carried the IUC3 virulence gene, additional long-read sequencing was performed with the Oxford Nanopore Mini-Ion platform, combining the short and long reads in a hybrid assembly using the Unicycler software ensured more complete and reliable genome reconstructions. Third, antibiotic susceptibility testing. All isolates were tested for their antibiotic resistant profile using broth microdilution method, which determined the minimum inhibitory concentration for each antibiotic. Testing followed Clinical and Laboratory Standard Institute (CLSI) guideline, and the minimum inhibitory concentration were interpreted according to European Committee on Antimicrobial Susceptibility Testing clinical breakpoints. This ensured that the data are comparable to other international studies and reliable for clinical relevance. Fourth, in silico analysis for genetic characterization. For genetic relationship and diversity, the team used core genome multi-local sequence typing (CGMLST), which is a high-resolution approach based on 2,358 core genes, and they also identified sequence type through the 7-gene MLST scheme, which help track the evolutionary relationship of isolates. Capsule and O antigen types were predicted using the captive tool to determine potential virulence factors linked to capsule polysaccharide. Fifth, detection of antibiotic resistance and virulence gene. The Klebsiella pipeline was used to screen assembled genomes for known antibiotic resistant genes and virulence associated genes like YBT, CLB, IRO, IUC, and RMPA or A2. Plasmid replicons were also identified to understand potential transmission routes of these genes, highlighting the role of mobile genetic element in spreading resistance and virulence. This multi-step methodology ensure a robust and comprehensive approach, combining culture-based technique with advanced genome sequencing and bioinformatics analysis. Such rigorous methods are essential for accurate surveillance of multi-drug resistant and virulent Klebsiella strain, providing a valuable insight into their epidemiology and potential health risks.
From the first results about population overview, the study analyzed 94 Klebsiella isolates, mostly from pigs, pork, cattle, and milk. 72 of these isolates, which means 77%, were part of K. pneumonia sensulato complex. Accurate species identifications was achieved using whole genome sequencing, correcting several misidentifications from multitoph. The second result of this study is about antibiotic resistance. Ampicillin resistance was found in 78% of the isolates. However, resistance to third-generation cephalosporin, fluoroquinolones, and colistin was low, and no carbapenem resistance was detected. Although 16 isolates had ESBL genes such as BLA SHV-27 and BLA CTX-M-15, only 5 showed resistance due to promoter mutations. For the third result about molecular typing, the isolates display high genetic diversity. 62 different sequence types were found among K. pneumonia isolates based on their DNA. 18 of these types were new. The most common type, called ST107, was found in only 4 samples. Other than that, in capsule typing, which is a layer that helps bacteria avoid the immune system, revealed 38 different KL types, with KL30 being the most frequent. The fourth result is about virulence genes. Virulence genes are the ones that make bacteria more dangerous, were found only in K. pneumonia isolates. Aerobactin was found in 19% and Yersinia bactin in 10% of the isolates. Aerobactin was exclusive to isolates from pigs and wild boars, suggesting a potential reservoir, meaning pigs might carry bacteria that are more dangerous and could spread to humans. Apart from that, no other major virulence genes like RMPA or CIB were detected.
Moving on to the discussion section, this study analyzed 94 Klebsiella species isolate from various animals and food sources to understand their genetic diversity, antibiotic resistance, and also virulence trait. One key finding is that species identification was not always consistent between traditional methods like multi-toph mass spectrometry and more advanced whole genome sequencing approaches. This underscores the superior accuracy of whole genome sequencing, especially in distinguishing closely related taxa like Klebsiella species. Notably, the majority isolates, 67 out of 94, belong to the Klebsiella pneumoniae sensu stricto group, and this suggests that Klebsiella pneumonia is quite widespread in the veterinary context, where it can act as a food colonizer or even as an infectious agent in livestock. Interestingly, the study also detected globally relevant sequence type, including sequence type 11 and sequence type 147, which are typically associated with human hospital infection. Their presence in animal isolates, specifically ST11 in pig and ST147 in cattle, hints at a possible cross-species transmission potentially from human to animal. However, the researcher did not detect any either hypervirulent or human-associated sequence type, suggesting that while some crossover may occur, it is not yet widespread. The genetic diversity among isolates was remarkable, with 62 different sequence type identified among the Klebsiella pneumonia sensu stricto isolates. Sequence type 107 was the most prevalent sequence type seen in 4 isolates, where 3 of them with capsule type KL10. While sequence type 107 has been found in some human cases, no clear link to a particular animal reservoir was observed here, and this broad diversity suggests that there is no dominant sequence type link to a specific animal host in the studied population. Regarding antibiotic resistance, the frequent detection of ampicillin resistance was expected because Klebsiella species naturally carry intrinsic beta-lactamase gene like BLA-SHV, BLA-OXY, BLA-OKP, and BLA-LEN. Extended spectrum beta-lactamase gene were found in 16 isolates, though only 5 carried BLA-CTXM genes associated with resistance to the third generation cephalosporins. Interestingly, the other 11 isolates had BLA-SHV27 and BLA-SHV41 genes with mutation in their promoter regions, which weaken the gene expression and result in a non-ESBL phenotype. This nuance is critical as it shows that while genetic markers for resistance exist, they do not always translate into the actual resistance in practice. A particularly rare finding was colistin resistant in a single sequence type 11 isolate from pigs. Unlike the typical MCR1 mediated mechanism seen in E. coli, this resistance was caused by an insertion in the MGRB gene. Such MGR disruption have been documented as the key drivers of colistin resistance in Klebsiella pneumonia. On the virulence side, only 22 isolates carried the IUC gene, which encode the siderophore aerobactin, which help the bacteria to scavenge iron in the host. Fascinatingly, all of these IUC3 positive isolates came from pigs and wild boar, hinting at a possible role for pigs as a reservoir of this potentially more virulent strain. These IUC3 genes were located on self-transmissible plasmid, suggesting that they can be shared across strain. However, the author emphasized that the further study are needed to confirm this hypothesis.
In conclusion, from this study, Klebsiella pneumonia was the most common type found in animals and food. Most bacteria were still treatable with antibiotics, but some showed warning signs. The presence of virulence plasmid, especially aerobactin in pig isolates, indicates pigs might serve as reservoirs of hypervirulent traits. So this study highlights the importance of continuous monitoring of zoonotic bacteria and resistance genes in the food chain. So that is all from us. Thank you.